Guys, so this is Roman numeral 4, neurons. A neuron is the cell that conducts our electrical impulses that the nervous system is based on. And so let's look at the anatomy of a neuron. So the first part, letter A, soma, that is the body. So another word for cell body is soma. And it's where you have the nucleus, it's where you have organelles, and where all basically the metabolic processes go on within a neuron. Letter B, the word Nissl bodies, which is here, are going to be clusters of ribosomes. So it turns out that, ribosomes create proteins, and neurons are really, really busy, metabolically active cells that create a lot of protein. And so they're going to have a very, robust population of ribosomes, both free in the cytoplasm, and remember ribosomes are on rough ER. So when you stain a neuron, it's going to stain very darkly. So that's what Nissl bodies are. Nissl bodies are just dark areas that pick up stain, which are ribosomes. Letter C, the axon hillock, is the region here, which is the origin of our axon. And we're going to have RER, so rough ER and Golgi, are absent in the axon, so proteins are made only in the cell body. They're transported via cytoskeleton called axon transport. So the proteins or the neurotransmitters that are made here, they're going to be traveling down, beginning at the axon hillock, traveling down, and then they're stored. Remember, the neurotransmitters are stored in our synaptic terminals there. So basically just axon transports them down. And then axons. So axons can be longer, they can be short. If they're rather long, they're called nerve fibers. So remember how we called muscle cells a fiber because they're just long and thin like a thread? So is the same as an axon. So for a neuron, the long axon is called a fiber. Okay, so it's called a nerve fiber. Sometimes these nerve fibers will branch. If they do, they are called collaterals, but they never branch as much as dendrites do, right? So if we look at our dendrites, which I actually left off, oh no, letter E is there, dendrites, way down at the bottom. So it receives signals and they're highly branched. Okay, so remember that signals or electrical impulses come into the dendrites, and then if a neuron wants to fire an active potential away, it'll fire it away down the axon. All right, so let me just show you, let's go back up to axons and just show you some collaterals. So here's a collateral, right? This is a collateral branch of an axon. The terminal arborizations is listed here. So you can see that the end of an axon will branch, and the branches, someone thought they look like a tree. So arborization is tree-like, right? The branches of a tree. And then at the very, very end, you're going to have synaptic terminals, also known as boutons. So those are the very ends of our axon where the neurotransmitters are stored, and that's what will secrete those neurotransmitters into a cleft to communicate with the next cell. So the next part of your outline, myelinated nerve fibers, non-myelinated nerve fibers. I went over this previously, so I'm going to skip that for now. And then there's something called a demyelination disorder. So before I get to that, let's just talk really quickly about this picture. So neurons, when they synapse with other cells, right, when they communicate with other cells, it's called a synapse. And neurons can synapse with another neuron, right? Neurons can synapse with muscles, so that's called a neuromuscular synapse, number 2. And neurons can also synapse with glands, so it's called neuroglandular synapse, right? But we'll look at a synapse at the end of this lecture a little bit carefully, but that's what we have here. This is a histology image of the neuron, so you can see that the cytoplasm of the body, of the soma, is very darkly staining, and that's due to those Nissl bodies. It's so dark that it actually creates a lighter staining nucleus. We can see some dendrites, these processes coming away from the soma, and then the one axonal process coming away from that body. Okay, so I'm not going to require this, but this is just a nice image of the neurons. And then all the cells in the background, so you can see a nucleus here, a nucleus here, a nucleus here, 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 here. Those would be those glial cells, right? Those support cells. And they outnumber neurons 9 to 1, so there are plenty of neural glial cells. So now let's talk about this demyelination disorder. So remember I said that myelin, the myelin sheath helps the nerve conduction proceed faster and it insulates the nerve fiber, just like how you have plastic around, your laptop chargers, your phone chargers, because the wires inside which conduct electricity are insulated and it makes the conduction, go unimpeded and uninterrupted. So if number one, multiple sclerosis. So this is a disease that it's actually considered a autoimmune disease, which means that your own immune system is going to destroy the myelin around your nerve fibers. So we can see this is a normal nerve fiber. We can see the internode, nice and robust and wrapped in concentric circles with myelin. Here we can see it's been damaged. And so we're exposing the nerve fiber and we're going to impede the flow of that impulse. So what's hard about multiple sclerosis in terms of diagnosis is that anywhere in the body you can have these episodes occur where you have demyelination happening. So if it were to happen in your brain, you can have cognitive symptoms. If it happens near the optic nerve, you can have visual disturbances. If it happens, in the body where the nerves go to your muscles, you have muscle weaknesses. So sensations, different sensations, bowel disruptions, urinary disruptions. So multiple sclerosis is really tricky to diagnose because when you go to the doctor and you might say to the doctor, I'm having blurry vision or I see spots or maybe my hand feels weak, I can't, make a fist, they're not going to automatically assume it's multiple sclerosis. They'll probably assume it's something else more common. So it takes a while to diagnose multiple sclerosis, but it is an autoimmune disorder. Okay, so varied symptoms because it just depends on where they happen in the body. And then number two, another cause of demyelination is heavy metal poisoning. This is why heavy metals are bad for you. So you want to limit your exposure to lead and arsenic and mercury because they actually damage those glial cells that create the myelin. And for pregnant women, especially with the developing fetus, right, they're not supposed to eat fish, these really large fish, because in those large fish like tuna or swordfish, they accumulate, it's called bioaccumulation, they accumulate mercury and lead that's in the environment. And so their bodies are actually pretty toxic. They have high levels of these heavy metals, and so pregnant women should not be eating them because those heavy metals, the percentage of heavy metals could affect the fetus. For us, being adults that are sort of already developed, the little bit of heavy metal is, it's bad. You're not supposed to eat a lot of these large fish, but if you do have tuna, it's not the end of the world. If you have occasional tuna sandwiches or occasional swordfish steaks, but you want to limit the amount. Okay, so I think that's it for the anatomy of a neuron. So let's look at the classifications of their shapes. So neurons have different shapes, and this might surprise you because up until now we've only been looking at this kind of cartoon of a neuron, right? We have a lot of dendrites, we have one long axon, and our terminal boutons or terminal arborizations with our boutons at the end. Actually, there are several kinds of shapes, so you're going to need to recognize them by how they look, or description, and know them by their name. So the one that we're most familiar with is called multipolar. And this is going to be a typical motor neuron. So any neuron that helps you move your skeletal muscle will look like this. Okay, this is a typical motor. Bipolar. So let's jump over here because bipolar is next in your outline. This is one soma in the center and then one really long process on either side, right? So one is going to behave like a dendrite, one's going to receive the information, one's going to behave like an axon and have information leave. But it's very difficult to tell, which one's which, kind of at a glance. So this is a pretty rare neuron. It's only in your eye and in your nose for smell. However, that's something you need to know. The next one is called pseudo-unipolar. So this one over here looks a little bit like bipolar, but it's not. You see how the cell body lays off to the side and you have a little bit of a stalk. And then you have an uninterrupted fiber that basically runs in both directions here. So Interestingly, they're both considered axons, but you do have a dendrite end, you do have the terminal end, and so these are going to be your typical sensory neurons. So imagine these are in your skin, then this will travel up. So if something touches your finger, this will travel up a nerve that's in your hand, up to your arm, it'll go to your spinal cord, right? And then this cell body is actually next to your spinal cord. And then from the spinal cord, this portion is going to travel in the spinal cord and then synapse with something so that the another neuron will take that message through the spinal cord up to your brain. Okay, so this is a typical sensory neuron. And then you have something called anaxonic neurons, where it's impossible to tell which one is the dendrite and which one's the axon because they're all highly branched. There's not a lot of information on anaxonic neurons, but these are only found in the central nervous system. Okay, so they're only found in the brain and the spinal cord. And they are responsible for making these interconnections. Because you can imagine being so highly branched, they can really make a lot of different connections to different interneurons. I kind of think of them as an operator, which can connect one incoming message to whatever neuron it needs to go to. Okay, so these are the shapes. Now let's look at their function. So let's move here. So neurons can be sensory neurons based on their function, right? So they either bring sensory information towards the central nervous system. They can be, letter B is motor neurons, so they're neurons that synapse with an effector, like a muscle cell or a gland. Or they are considered an interneuron, which is something that only makes those interconnections within the central nervous system. Okay, and these are the most populous. They are 99% of all the neurons in the body. So these are the three categories of their function. The neurons are either sensory or they're interneurons or they're motor neurons. And just take a look at the shape, right? Remember this shape? We have a cell body off to the side, we have a stalk, we have a very long fiber. So remember, let's try and take a guess. If you said pseudo-unipolar, you are correct. What is the shape of this? This is your anaxonic shape. And if you look at this, right, our motor neuron, what shape is the motor neuron? This is a multipolar shape. Okay, so let's go to our question. Sensory neurons of the peripheral nervous system, which may be myelinated, are usually. All right, so hopefully this is easy. Sensory neurons are pseudo-unipolar, right? Okay, so that's your answer. All right, so let's look at nerve impulses. So an impulse, of course, is an all or none event, which means that the neuron is either firing an action potential or not. So there's no such thing as a large or small action potential. There is only one kind of action potential. Okay, so there's no big or little impulse. So your messages that get to the next cell, how are they mediated, right? How can you tell a big message versus a big stimulus versus a small stimulus? So that's 1 and 2. Oh, wait. No, I don't have it here. So basically it's based on how fast the impulses come. So if you have many, many impulses coming into a receiving cell at one sort of segment of time, that's going to be a stronger impulse. If you have fewer impulses coming through and fewer neurotransmitters being secreted, that's going to be a smaller stimulus. Okay, so that stimulation is based on how many impulses come, not if it's a big or small impulse, because there's no such thing as a big or small impulse. Okay, and then let's just look at the speed of conduction. So we know, number one, that how fast a message gets one place to another is whether the axon's myelinated or not. It's 100 times faster if it's myelinated. And also the diameter. So it turns out the diameter of the axon has a role. If it's a large diameter, the speed, it travels faster. Okay,